just kick off with the presentation. So today we have two presentations from Evie and Amrita, and we will start with Evie. So yeah, you can go. So Evie completed a bachelor in psychology in Dublin in 2021. And then she did a master in clinical neuroscience here at the University of Galway in 2023. And now she started a PhD in psychiatric genomics in the Department of Biochemistry. And her research focuses on exploring the role of genetic in social participation and occupational engagement. And I believe she will tell us a bit more about that now. So thank you, Evie.
Thank you. Thanks for that lovely introduction. So just to introduce myself for anyone that doesn't know me, my name is Evie Doherty. I'm a third-year PhD student within the School of Biological and Chemical Sciences here at the University of Galway. And for today's IHDI seminar, I'm going to just be discussing my most recent work, which is a genome-wide association analysis of social participation and occupational function within the UK Biobank. So this research was carried out under a wider research team referred to as SOGEN. So our aim is to basically carry out research on genetics, environmental and neuroimaging biomarker factors that underlie social and occupational functioning. And my area is obviously under the genetics. So this research was carried out under the primary supervision of Professor Derek Morris, who is the head of a school in the biological and chemical sciences. Also Professor Gary Donohoe, who is within the School of Psychology, and Professor Dara, who is based within the School of Medicine. So just to give you some insight on what you can expect in today's seminar, so I'm going to be discussing psychosocial disability. I'll give you some insight into what that is, and then what it looks like in a psychosis cohort. I'm going to discuss the study aims and objectives, the methodologies, or rather the approaches that we took to carrying out this research, the findings, and then I'm going to just tie it all together and draw some conclusions from our findings for all of you. So just to begin, what is psychosocial disability? So psychosocial disability refers to the social and occupational challenges that are associated with mental ill health. These challenges are in fact considered distinct from psychiatric disability, which encapsulates clinical impairment. So clinical impairment is generally what would lead to a neuropsychiatric diagnosis for someone. And for this reason, psychosocial disability is very often overlooked. So psychosocial disability is quite prominent in a whole range of neuropsychiatric disorders, but one such cohort that are particularly affected are those who are living with psychosis. So psychosis is generally characterized by hallucinations and/or delusions. So for anyone who doesn't know, hallucinations are essentially hearing or seeing things that are not there, and delusions are essentially the belief in a false narrative. So while these are characteristic of psychosis, impaired social participation and occupational function, which are essentially umbrella termed as psychosocial disability, are also key features within psychosis. Psychosocial disability has been associated with both the development and the trajectory of psychosis, in that those who have greater psychosocial disability are more likely to have a worse trajectory throughout the course of their illness. Individuals actually report that psychosocial disability is the greatest burden in which they experience throughout the course of their psychosis, with around 50% of individuals actually failing to regain pre-morbid psychosocial functioning even during periods of remission. So when I say remission, I'm generally referring to non-active phase symptoms, so whenever people are essentially stable within their disorder. Despite the fact that it is so prominent within psychosis cohorts, as well as other neuropsychiatric disorders, psychosocial disability is not very well understood and our understanding is quite limited. So that's where our research comes in. So what were the aims and the objectives here? So the aim of my research in particular was to gain a better understanding of the genetic underpinnings that underlie psychosocial disability. We strive to conduct a genome-wide association study in a UK Biobank sample to identify the genetic variants that are associated with social participation and occupational function specifically. Social participation and occupational function are two phenotypes in which we derived ourselves using UK Biobank data, and I'll go on to explain these in a little bit more detail further along in the talk. So now moving on to our methodology. What approaches did we take to carrying out this research? So what we did was we used a UK Biobank sample. So for anyone that doesn't know, the UK Biobank is a large-scale biomedical database that contains data for around 500,000 individuals. What we had was a maximum sample of 404,569 of these UK Biobank individuals, all of whom were aged between 39 and 73 years, and where there was a greater proportion of females to males. These individuals were all recruited from around 23 assessment centres from across the UK. As you already know, we carried out a genome-wide association study, which might be better known to some of you as a GWAS. So essentially what this is a series of regressions where we look for a relationship between genotypes and our phenotypes of interest. I'll give you a little bit more detail on this further along too. So what we did here was we used a contemporary method, which is referred to as fast GWA, and this allowed us to maximize our sample size by retaining related individuals who would typically have been removed in the more traditional methods. And then we also carried out a whole host of post-GWAS analysis, including SNP-based heritability, gene set analysis, gene mapping, gene prioritization, TWAS, as well as genetic correlations and Mendelian randomization. So as promised, I want to provide you a little bit more information on the phenotypes that we used. So for social participation, we define this as the active participation in social activities and our social interactions. So our aim for this variable was to basically capture how well someone actively and outwardly functions within society, rather than how well they feel that they were functioning. What we did was we took 2 UK Biobank questions from the available questionnaire data. These were number of leisure activities and the frequency of friend and family visits, and we very simply added these together to create a composite score that ranged from 0 to 10, with greater scoring meaning greater social participation. For occupational function then, we defined this as the extent to which an individual actively engages occupationally or vocationally. And the idea behind this variable was that we could capture how well someone was actively and outwardly functioning in terms of their occupation and vocation, but not just whether or not they were employed. What we did was we took the current employment status variable that was available within the UK Biobank and recoded it so it ranged from -2 to 4, with greater scoring, once again representing greater occupational functioning. So just for anyone that does not have a background in genetics, I'm going to discuss a little bit what genome-wide association studies are. So basically what it does, it allows us to look for associations between millions of genotypes with specific phenotypes, which in our case was social participation and occupational function, in one large analysis. So because there's millions of genotypes, you're essentially carrying out millions of regressions, but all within one single analysis. The genotypes in this case are single nucleotide polymorphisms, which may be better known to some of you as SNPs. And these represent a difference in a single DNA building block within the genome. So just to give you an idea, where I may have an A, you may have a G, and the person next to you may have a T at that exact same position. But because I have an A at that position, I might be more susceptible to developing a certain trait, a certain disease, or a certain disorder. However, a single variant or single SNP does not necessarily have this much penetrance, but rather it is a group of SNPs that could make you more susceptible to a certain trait, disease, or disorder. But just to simplify it for your understanding, I have this here. And then because you're running millions of regressions within a single analysis, there must be a p-value corrected threshold. And in this case, this is the widely accepted p-value corrected threshold for a GWAS, and this is 5 by 10 to the -8. Okay, so now just to get into our findings of this study. So for social participation, we find that there was 18 lead SNPs or genetic variants across 17 genomic regions, which are better known as loci. There is a SNP-based heritability of 4.1%, meaning that for the variation that we observe in social participation, SNPs can account for around 4.1% of this variation. Then for occupational function, we find that there were 3 lead SNPs or genetic variants at a single genomic locus. and that there was a SNP-based heritability of 1.8%, meaning that for the variation that we observe in occupational function, SNPs within the genome account for around 2% of this variation. So in the grander scheme of things, it is quite minimal, but nonetheless, it is an effect. So moving beyond the GWAS results, then we carried out a whole series of post hoc analysis, as I said. So the first of these was the gene set analysis. So this essentially allows us to take 10,500 in and around 10,500 hallmark gene sets that are responsible for different processes within the body and look to see if there's any associations between these gene sets and our phenotypes of interest. What we found here was that the gene set responsible for the regulation of synaptic structure and/or activity was significantly enriched for genes that were associated with social participation specifically. This was quite interesting to us because such pathways have also been enriched for genes that are associated with psychotic disorders such as schizophrenia and bipolar, basically implying that there might be some sort of relationship here between how well we engage actively and how genetically predisposed we are to these disorders. Then we carried out gene mapping and gene prioritization. So just to give you some context so you can maybe understand what gene mapping and prioritization is. So gene mapping is where we take the variants that we find within our GWAS and link them to genes based on the location of those genes to the variants that we found. We carried out positional mapping, 3D chromatin mapping, and EQTL mapping, as you can see here. So positional mapping is essentially where the variants are identified, how close they are in proximity to that gene. So the association is through the proximity of the gene. 3D chromatin then, chromatin is basically structured in a 3D manner. So the location of a particular variant may affect the gene further downstream or further upstream based on the structure of that chromatin. And then lastly, the EQTL is where we take gene expression data and make associations between our variants and the expression of that gene. So does a variant increase or decrease the expression of a certain gene? Then you had MAGMA analysis, which is one step above mapping. So this basically allows us to take not just a single SNP, but a grouping of SNPs and to see if these groups of SNPs are associated with particular genes. So just having provided you some context on that, to show you our findings, then we find that across all four of these mapping approaches and MAGMA, we found that there were 3 genes that were consistently mapped to social participation specifically. And these were CSE1L, TNRC6B, and STI1. These genes, 2 of these genes, which are CSE1L and TNR6B, were also prioritized by the gene prioritization approach, which essentially allows us to identify causal genes of functional importance within social participation. So are these genes, a change in these genes, causal for good or bad social participation? So given that these genes were consistently mapped and prioritized, I'm just going to provide you a little bit more context on them. So CSE1L is involved in cell proliferation and apoptosis. It is a target gene for microRNA 137 and therefore is a well-established schizophrenia risk locus. And also increased expression of this gene has been associated with an elevated risk for schizophrenia. Then for TNRC6B, which was the second gene consistently mapped, this enables RNA binding activity and is involved in translational inhibition. It has also been previously linked to intellectual disability, autistic traits, ADHD, as well as subsequent behavioral abnormalities, all which have been previously associated with social issues. And for occupational function, although the findings are not as interesting, it's always important to show them anyway. But you can see that we use the same approaches here. So we found that while there was no gene consistently mapped across all approaches, there were 6 genes consistently mapped across positional 3D chromatin and EQTL, which you can see just down here. The gene prioritization approach then prioritized 2 completely different genes, which are circled here just at the bottom of the screen. Nonetheless, there are still some genes I'd like to point out for you. So in the MAGMA analysis, DRD2 was essentially mapped to occupational function. So this DRD2 encodes for the dopamine D2 receptor. It is the target of all currently effective antipsychotic drugs, and it has been consistently linked to schizophrenia. Then in terms of our mapping approaches, the most consistently mapped was T-SNARE with the greatest significance. And this belongs to a group of proteins which are believed to increase the schizophrenia risk by synaptic dysfunction. There's not a lot of information on the role of this gene other than this, but it was quite promising that we found even this. So just to draw some conclusions on our research, so we can say that there is a significant genetic component to psychosocial disability. Albeit low, that there is these traits or phenotypes are actually heritable. We can confirm that there are shared genetics with neuropsychiatric disorders as well as other health-related traits. And there is some evidence of causal relationships. So just to highlight the strengths and the limitations of the study. So we had a large sample size, which was facilitated by FAST-GWA, which essentially allowed us to retain related individuals. We were also able to derive active functioning measures, which allowed us to measure people's outward and objective functioning in the absence of subjective influence. Then the limitations, there are some suitable because we were looking for active outward functioning, we were limited in terms of the suitable variables within the UK Biobank. So this was a limitation for us. Also, it's limited generalizability because we used those who are of white British ancestry only. And also the inferences that we draw from this research should be taken with care, as this is a cross-sectional research project. So I'd just like to say thank you to everyone here today for listening. And also to my principal investigators, in particular Professor Derek Morris, and then of course everyone from the wider SOGEN team. Thanks.
If anyone has questions. Oh, yeah.
Some question. Was there any specific excluding criteria for did you include anyone who actually provided the
Essentially anyone that provided the answer we included, just so that we could get a very rounded idea from a general population. Yeah.
Psychosocial disability is like a questionnaire that the donors of these samples are filling out?
Yeah, so in the UK Biobank, whenever people are recruited, they are given a questionnaire with a series of questions that cover a whole range of domains, and within that questionnaire, they fill out those questions. But that's the problem, that it's optional whether or not you want to fill out a certain question, so that's why it can be a little tricky with our sample sizes.